If you want to do it by yourself, feel free to do that as well. So with that out of the way, I did make a little boo-boo last class. I did miss a little bit of something about how things move in and out of bacterial cells. So we talked about the cell wall, we talked about the S layer, we talked about the capsule, but we didn't really talk about how things move in and out of the cell. And in particular, how do things, principally chemical signals, get through the cell wall with all of that peptidoglycan in the way? And the answer to this question is the fact that the cell wall is not really a wall in the the sort of the standard way we think of a wall, right? It's not like the wall that was behind me when I had my face camera on. It's a wall as in it's porous. It's more like a sponge. And so the bacterial cell wall, again, it's wall is it's not the right word for it. It's more like a sponge, frankly. Things pass right through it. You can also think about it as like Jello. So let's look at the structure. We'll see why. So we have the sugar backbone, which is the nag and NAM repeats, as we discussed about. And this is in orange here, and this is what it looks like as molecules. And then we have the green, which are the peptide links. Remember, peptidoglycan, which is made of those nag and NAM repeats, are joined together. So this, for instance, these two repeats would be joined together vertically. But horizontally, we have those nag and NAM repeats. Now, the interesting thing is when you look at the shape from the top down, C, it looks like the Mercedes logo. At least that's the way you could look at it if you like cars, I guess. This is the way it looks like, right? But if you look at it from the top and it looks like the Mercedes logo, that means there's that inner part here that's solid, and you can see that here, but that means there's little pores around it. And in fact, that's actually what you see. And so what we see for peptidoglycan, this would be a top view of what peptidoglycan would look like, and they orient themselves in a net-like shape. The idea being is that the points of the stars are facing each other, and this is what we would look at. Right? This is what peptidoglycan looks like. It's essentially like a fence, and we look at the molecular structure, and this is what it looks like. It looks like a chain link fence. And now thinking about how things would move in and out of there, well, I hope this is clear as to, well, it's not just a wall, right? It's not like trying to pass something through a brick wall. Again, this is like trying to move something through like a cheesecloth or a filter or like a sponge, right? Things are going to move through because there's pores. And just as a note, there has to be pores, right? Bacteria have to be able to interact with their environment. They have to be able to uptake food. They have to be able to uptake nutrients. They have to be able to excrete things, right? They need to be able to move things in and out. So there has to be porous layers. And this is not just true for bacteria. This is true for every organism, right? Whether you're a bacteria, an archaea, or a human, you have to move things in and out of yourself. So you can't be completely shut down. But these openings allow the passages of small molecules through the cell wall. But one of the things that they do prevent is the passages of large molecules. And so when we think about the cell wall, we want to think of things, this is like a sieve, right? If you've ever been to the playground when you were a kid, you had a sieve for the sand, and you sieve the sand through those, your sieve, and has those tiny little holes. Only some of the things made it through your sieve. And that's the same thing with the cell wall for a bacteria. Only some things make it through. So very, very, very large things. Think of something like a virus wouldn't just pass through. Something very large, like a very large complex sugar, wouldn't just make it through. But something very small, like say glucose, or maybe you have a calcium ion, those things could move just right through. But there is a little bit more. And so one of the things that they do have are these what they're called ACR, AB, Toll C complexes. I know that sounds like it's made up. And frankly, some of science does kind of sound made up, but that's just the way science is. But these are efflux pumps that move small molecules, toxins, other large things out of the cell. And actually, one of the interesting things about Toll C and our ACR-AB complexes is that they are really, really important for antibiotic resistance. So we're going to talk about antibiotic resistance later in the semester, but this is sort of one of the sort of early introductions to it, is that these receptors, these proteins that span both this would be a gram-negative bacteria, right? Because it has the outer and inner membrane and a very thin layer of peptidoglycan. What we're seeing here is that these are spanning both of them. So what they're doing is they're pumping things from the inside of the cell to the outside of the cell. How does it get through the cell wall, you could ask? Well, it goes through one of the pores. So the cell wall, which would be about here, it is absent from this figure, just the way they drew it, but is poking through one of those holes. The toll C is the purple thing, and in the other side, it's the teal. Um, it's poking through this. It's sitting right on top of the peptidoglycan, so it's sticking out through the membrane, basically poking its way through these pores. So for things that are too big or too toxic to move directly through a cell wall, a toll C ARA pump will push it through and push it out of the cell. And now there's also, just as the one last note, there's also the reverse of this. So this complex moves things out of the cell. There is complexes that move things into the cell. And we'll discuss that later in the semester, how things move in and out. But just wanted to make note of sort of how the structure of the cell wall relates to the movement, what we would call active movement of large or potentially bad things from a bacterial cell. So fun fact. And then I guess the other thing I should make mention is the size here, right? We have 100 angstroms, very, 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 very tiny measurement. So we're talking very, very small. We're at the molecular scale here. So again, 100 angstroms, if I'm not mistaken, is 10 to the minus 9 meters. So it's very, very small. So tiny, 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 tiny. So with that out of the way, let's talk about microbial structure and morphology. So what gives a microbe its shape and how are morphologies useful? What structures do prokaryotes use to help them move and attach? And how do prokaryotes use chemical gradients to direct movement? And this last one is going to, you want to keep this last one in the back of your mind for an extended period in this class. And I say this not because it's a particularly foundational thing, but we're going to talk about gradients later in the semester when we talk about metabolism. So it's a pretty important topic that you want to keep in the back of your mind for, I think, lecture like 8. I think, or 8 or 7, the lecture everyone's going to hate because everyone always hates metabolism lectures, and it's just the way it is. But metabolism all is based upon chemical gradients. So let's dive in. So cell membranes as a whole provide a selective barrier. And the cell walls and the S layers give microbes structure, but what gives a microbe its shape? And what gives a microbe its shape, very much like what gives your cell your shape, is actually this what's called the cytoskeleton. And this is exactly what it is. Cyto meaning cell, skeleton meaning skeleton. It's essentially a skeleton of a microscopic organism or of a cell, right? And so that's what a cytoskeleton is. And it's not made out of calcium bone like ours, but it's made out of typically proteins or sugars. So when we're thinking about our prokaryotic skeletons, there are 3 key proteins that we need to discuss. So the first is FTSZ, which is a homolog of tubulin. For those of you that have heard of tubulin before, you're probably thinking, oh, where do I know that from? Well, you should know that because that's part of your cytoskeleton. Your cytoskeleton is made of tubulin. And I say your cytoskeleton as in the skeleton of your cells, not your body skeleton. And what this does is it forms a Z ring, which is essential for cell separation and division. And you can see a little diagram here. This is a ring of FTSZ around the center of this rod-shaped bacteria. So that's the first one. The next one is MREB or REB. It's the homolog of actin. Again, should come to mind, actin, part of your cytoskeleton as well. It's also the main component of muscle fiber, which allows muscles to be very, very elasticy and stretch. What this does is it forms a coil inside the cell and allows the cell to elongate. So cells that are rod-shaped or bacilli, it allows these cells to elongate. And then finally, we have CRES or CRES, which is the crescentin protein. It has no cool homolog like tubulin or actin, but it is the homolog of intermediate filaments, which again are part of your cytoskeleton, but not particularly very exciting. And what it does is it actually curves the inside of crescent-shaped bacteria. And so I think before we sort of move on, I just want to talk about the evolutionary sort of significance of this, right? We talked about the origins of eukaryotic life last class, right? And we talked about how eukaryotes are more closely related to archaea than they are of bacteria. But these 3 proteins are pretty bacterial-like are pretty confined to the bacteria. Now, they are found in archaea, but they're found pretty heavily in bacteria. So what it implies is that this cytoskeleton structure evolves early and was essentially passed down from bacteria, the ancestor of bacteria and archaea, to both bacteria and archaea, and then archaea then pass it down to eukaryotes. So I think that's actually a pretty kind of cool thing to think about, right? This is sort of classical evolution, right? Descent with modification. It's a homolog, meaning it's very similar to our tubulin, but it's different than tubulin because evolution has changed it to benefit eukaryotic cells. So just a fun fact. I think this sort of the parallels here are pretty astounding from an evolutionary perspective. It's if we sort of looked at this, much more in depth, you could see all sorts of interesting things. But we're going to move on for the sake of discussion. Now, let's talk about MREB or MREB. And so it's present in bacteria, and it's related to eukaryotic actin. It was actually identified because those that were mutants in MREB, meaning they lacked the capacity to express the MREB protein, they actually couldn't elongate and had odd spherical shapes. So this is what a normal bacteria with MREB looks like. It's rod-shaped, pretty common bacterial structure. This is 5 microns, so it's a pretty big bacteria. Most bacteria, as we mentioned, are about 1 microns. And that's in contrast to MREB mutant. And it forms these weird globules. So I hope it's pretty clear what the effect of MREB is, right? It's pretty dramatic, right? And it has a direct effect on the shape of bacteria. And what it does is it forms a helical structure below the cytoplasmic membrane, and I'll show you what that looks like in a moment. But it does allow cells to elongate, and is essentially found in every rod-shaped bacteria. As we'll discuss in lab, and we'll discuss in the lecture as well, there's a pretty limited number of shapes bacteria take, and one of the most common shapes bacteria take is rod-shaped. Rod-shaped is by far the most common shape bacteria take. So let's look at MREB real quick. Oops, sorry about that. And let's look at all these other proteins. So we'll have 3 bacteria here. We have Staph aureus. So if you've ever had a staph infection, it's caused by this jerk. If you've ever heard of MRSA or methicillin-resistant Staphylococcus aureus, it's this jerk as well. We have E. coli, which you guys have all heard of, whether it's from an outbreak of E. coli, like from Chipotle, or you just heard of it because it's a model organism. And then finally, we have Caulobacter crescentus, which is a waterborne pathogen, but it's also just found in water as well, but it can cause some disease. And so we have FTSZ is involved. It's a ring-like structure around the center. Again, it helps with budding here, so separation from one cell into 2. We have MREB that again acts as like a coil. It helps with the elongation. And then finally, we have crescentin, which I think it's kind of interesting with Caulobacter because it has all 3. So it has FTSZ, which again has that budding behavior. It has MREB, which has that coil behavior, so elongation, and bacillus or rod-like shaped. And then finally has crescentin, which gives it that curve. And we'll talk about the benefits of that curve for Caulobacter in a few minutes. But these are our 3 proteins. And we're talking about these proteins directly because they are very important for cellular morphology. Now, why the heck do we care about morphology, right? Why do we care about things, the shape they take, right? Well, you should know that shape matters. What you look like matters, right? What animal looks like matters, right? You don't see a turtle running 60 miles an hour, right? You see cheetahs running 60 miles an hour because they have the anatomy to do that. But turtles don't have the anatomy to do that. So morphology, structure is functional. This is not just true for animals. This is true for everything. Structure is directly linked to function. And I'm just gonna put this out there because we're gonna talk about it again later in the semester. But this is true from the biggest levels you can think of, so organismal levels, all the way down to the molecular level. Structure and function in biology are directly related to one another. And this has an underlying basic principle in chemistry, but just to know, structure and function are linked together. Now, let's talk about size in the microscopic world. So size differences are pretty stark. Size differences are pretty stark. As we talked about, viruses are, we have a comparison here. We have eukaryotic cells, which are pretty big. We have viruses that are tiny, and prokaryotic cells, which are still pretty tiny, but still much bigger than viruses. So we can look at the average sizes. Our prokaryotic cell is an average size of 1 to 3 microns. Eukaryotes range 15 to 20 microns. This is not like human cells. This is like protists, fungi, things like that. And then we have our viruses, which are about 20 to 200 nanometers. So very big differences in size, right? And the other thing I'd like to make mention of is this prokaryotic cell size is for bacteria in clinical settings, or prokaryotes in clinical settings. We do know that there are environmental bacteria out there that are much, much smaller. So just as a note. Now, when we think about the advantages of being big, well, it means you're less likely to be eaten by something, right? Something that's big like a tiger is less likely to be eaten by something in nature. Just give me one sec. I need to let my dog out. I apologize for the break. Give me one second. Sorry about that, guys. I love the dogs, but man, are they a pain in the butt sometimes. All right, so back to size differences. So as I mentioned, being big has advantages, right? Less predation means you're less likely to get eaten. Right? Tigers aren't getting eaten in the wild, but smaller things are, right? You think about a bird, think about a mouse, right? Those things are getting eaten. Same thing for bacteria. Same thing for microscopic eukaryotic organisms. The smaller you are, the more likely you are to get preyed upon. In addition, being big allows for the more stuff, whether that is nutrients, water, or the presence of organelles. The advantages of being small, it means there's less upkeep to your cell, it means you can reproduce faster, and it means you can take up nutrients and other food faster. So there is costs and benefits to being small and costs of benefits of being large as well. But morphology, as I mentioned, is functional. And it's really important in bacteria because that small size is important for them. Right? If you're a pathogen, that small size is important for you. It allows you to evade the immune system. But size overall, whether you're a pathogen or an environmental microbe, is important. And what bacteria end up having is they have a very large surface area to volume ratio. And what does that mean? Well, let's take a hypothetical bacteria here. It's 1 micron wide by 5 microns long. We can use math and calculate the surface area of a rod. It gives us to 37.7 microns squared. Then we can calculate the internal volume at 15.7 microns cubed. So why does that matter? Why does having more surface area to volume matter? Well, what physics tells us is by having that ratio be in favor of surface area to volume, it allows for the rapid uptake in diffusion, so movement of things from a concentration of high to low, for diffusion of things into the cell and the excretion of waste out of the cell, or the excretion of anything out of the cell. If this ratio was flipped, if bacteria had a higher volume than they did surface area, they would not be able to uptake and excrete things as fast. And actually, what we actually see for eukaryotes is this. We see a higher volume than surface area, which means that in eukaryotes, they uptake things actually less fast. This was a cell biology course, we talk about that a lot more, but just to know the situation is flipped. So bacteria, it's a very high surface to volume ratio, and in eukaryotes, it's a very low surface to volume ratio. So that's the first part. Again, rapid uptake is important. Think about this in any context, right? If you're a pathogen, being able to respond rapidly to your environment is important. But if you're just a single cellular organism, that's important too. You need to be able to respond rapidly, and that's one of the main characteristics bacteria have. And so that's our surface area to volume. Another part of morphology, again, is functional, but is this idea of cocci versus bacilli. So these are the two most common cellular morphologies we see in bacteria, cocci being round and bacillus being rod-shaped. As a general rule, if you're bacillus, you're likely to be motile, meaning you have the capacity to move. If you are cocci, you're likely not able to move. But there are exceptions to both rules. There are bacilli that are incapable of movement and cocci that have the capacity to movement. But as a general rule, if you're round, you're not swimming. If you're rod-shaped, you're swimming. And some organisms have the capacity to take on different morphologies depending on the situation. And these organisms are what we call pleomorphic. And what do I mean by that? Well, we're going to talk about one of the cooler bacteria out there. This is Helicobacter pylori. If you've never heard of Helicobacter pylori, he has a really interesting story. This is the causative agent of ulcers. So if you've ever known anyone with an ulcer, well, the reason that person had an ulcer is because of Helicobacter pylori. It's a really, really fascinating bug. It lives in your stomach. It embeds itself on your stomach lining. It is a natural part of your gut microbiome, but it can cause some pretty serious infections if you have the right strain of Helicobacter pylori. So, but it's kind of an interesting thing. When we look at Helicobacter's morphology, it has this pleomorphic life cycle where it has, um, when it's trying to infect a host, it is elongated, so it's bacilli shape, and is motile. So this is during early infection and sort of in the middle of the infection. It has a completely different shape relative to late infection. As the infection is starting to go away, um, it becomes round and non-motile. So it has this complete shift in its morphology between early infection and late infection. And this is what we call pleomorphic. And it is functional. And how is it functional? Well, for Helicobacter pylori to do its thing, it has to physically get into your stomach. And once it's in your stomach, it needs to be able to make it to your stomach lining. It can't do much sort of swimming around in the juices, the acid, the liquids you've drank in that are in your stomach, but it needs to make it to your stomach lining. And so for it to cause infection, for it to be part of your stomach, it has to swim through all your gastric juices and embed itself into your cell lining, your stomach lining on the edge of your stomach. And it has to be motile to do that. So it is beneficial for this bacteria early in infection to be motile so that it can physically swim and then infect you. But later infection, it doesn't really matter, right? Later infection implies that it's going down. So let's move from structure. Structure also, as we've mentioned, is involved in prokaryotic mobility. Now, mobility or motility in a prokaryotic organism is largely reliant on what we call passive movement. And what do I mean by passive movement? Well, if I have a bacteria in the ocean and that bacteria moves from point A to point B because of the ocean's tides or the currents, that's passive movement. If I have a bacteria that's going through my colon because it's hitching a ride on my poop, well, that's passive movement. So the vast majority of prokaryotic movement is reliant on passive movement. And the other big one in terms of passive movement I should make mention is this random movement in liquids, which is called Brownian motion. And so liquids, if we were in person and we were looking at microscopes together, you would see this. But Brownian motion is just random motion of water, just due to just the random nature of how water is binds to one another and stuff like that. But just to know, there passive movement is not just like currents and poop and stuff like that. It is actual like Brownian motion. So just as a note. But bacteria do have the capacity to have targetive movement. And this is commonly done by a flagella or a flagellum or flagellum, depending how you want to say it. And flagella are long spiral-shaped rods that stick up from a cell surface. And so we can look at this over here. And there's actually, the reason I like this sort of triple pane of figure, because we have a couple different types of morphologies here. So we have a single flagella, we have 3 flagella, and then we have this crazy bacteria here that's got all the flagella in the world sticking out from the side. So this is sort of the sort of the most common types of flagellar orientation we see on bacterial cell walls. And these are hooked onto cells by a complex of proteins that allows for movement. And so we're going to first talk about bacterial flagellar movement, and then we'll talk about archaeal flagellar movement. It's a tough word to say, apparently, because they are different. So we're going to talk about both. And so each flagellum is a spiral filament of proteins, in particular protein monomers called flagellin. And it's a filament that's rotated by a motor driven by the proton motive force. Now, proton motive force should ring a bell from intro bio, but proton motive force, we're going to talk about this later in the semester as well, but this is the primary mechanism by which cells generate energy by respiration, so proton motive force, just keep that in mind. The idea being proton motive force is like, you could think of it like a dam, right? A hydroelectric dam. You have water flowing in, it turns a turbine. That's what proton motive force is. Think about it that way. But the idea being, for bacteria, as protons flow in, the flagella rotates. And the crazy thing about the flagella is it can rotate anywhere between 18,000 and 100,000 revolutions per minute, which is crazy, right? That's 100,000 rotations per minute. Think about like a jet engine, right? A jet engine spins at 100,000 times per minute. That's crazy fast. And so it does allow bacteria to move incredibly fast. So relative to their size, it's very fast. If you scaled it up, it would be the equivalent of running 40 to 200 miles per hour. So really, really fast. And actually the cool thing is the fastest bacteria on the planet is a bacteria that lives in marine environments. and it can actually swim 750 microns per second. Now that doesn't sound like a lot because it's not, right? We're almost to a full millimeter per second, but that is 750 times the length of the bacteria's cell. Which, to put that in perspective, if I swam 750, or if I ran 750 times my height, that'd be 6 feet times 750, which would be about what? Uh, I can't math, but something really big. What, 3,200 feet per second? Really, really fast. Half a mile a second. Really fast. So these guys are cranking there. But with that out of the way, with the craziness of flagellar movement out of the way, their motor does possess what we call axial and rotary parts, and I'll show you a picture of that in a moment. And they're composed of specific proteins. And the two key proteins we're going to discuss are MOTB, or MOTB, and FLIG, or FLIG. You know, I'll say one thing about people that name proteins. They always pick the kind of the funky sounding names, but MOTB does form the part of a proton channel, and FLIG-G, or FLIG, forms part of a device that generates torque. And just as a note, flagellar movement is either the flagella spinning clockwise or counterclockwise relative to the cell. Just keep that in mind. It's going to become important. Now, let's take a peek here. So we have FLIG-C, which is, again, our flagellar protein here. We talked about this. I mentioned this one a few slides ago. That creates the actual flagella, again, that whip-like structure. So if this is a cartoonized version. This is what an electron microscope picture from some people at Brandeis University looks like, just to know that there is a correlation here. Then we have the outer membrane, and then we have the peptidoglycan, and then we have the inner membrane. For those of you that are keeping track, this would be a gram-negative bacteria, right? No thick layer of peptidoglycan. Embedded inside the outer membrane, we have the L ring. Again, this is all just a giant protein structure. That's really all it is. We have the P ring embedded in the peptidoglycan ring, and then we have the MS ring, which is embedded in the intercellular membrane. Now, that's our sort of anchor, right? It has to anchor to that cell wall, and it has to anchor to that cell membrane. But then we have our two proteins that are important. We have MOTB, and we have FLIG-G. Now, they're connected directly to one another. So we have MOTB, which is connected to MOT-A, which is embedded inside the inner cellular membrane. But you'll notice that MOTB is transferring protons in from the sort of this periplasmic space here into the inner cytoplasmic space. Now, why does that matter? Why does transferring of protons matter? Well, what we will talk about later in the semester is moving protons, sending them through proteins, actually acts as a way of generating energy. This is what we call proton motive force. A hydrogen ion is just a proton, right? H plus is just a hydrogen missing its electron. By sending it through these proteins, what it does is it generates energy. What it's generating for energy is ATP. By generating that proton motive force, it's then passing that energy to FLIG-G, and by passing it to FLIG-G, it's causing the spinning of the flagella. And that's the basics of this system. MOTB creates a proton motive force, creates energy, spins FLIG or FLIG, and then causes the flagella to spin. And the flagella can do a couple different things. It can do a forward run. It can cause the bacteria to tumble. It can cause a forward run or a tumble. So typically speaking, counterclockwise causes a forward run. Clockwise causes what we call a tumble. So you can sort of see that orientation. You know, if you had a clock, you know, here, counterclockwise goes this way. That's why this is spinning, causing a forward run. Clockwise spinning causes a tumble. And so just as a note, this is our gram-negative. Let's see what the differences here are for our gram-positive versus gram-negative. So this is the one we've talked about. But you'll notice that it is a pretty heavily conserved structure, right? The outer flagella is pretty similar. The proteins are similar in shape, but they have different overall structure. So FLG is our gram negatives, and FLH is our gram positives. But you notice that our gram positives also have FLG as well. So it's very, very similar, a very, very conserved structure as well, but slightly different between gram positives and gram negatives. But you'll notice that the proteins that anchor the inner membrane to the peptidoglycan cell wall are similar, and the internal guts of these are very, very similar. So what this tells us is that this structure, this flagellar movement, evolved before this in evolutionary split between gram-positive and gram-negative bacteria. But very, very similar, just a different way to anchor it. A slightly different protein composition, but ultimately they're doing the same thing. So that's our bacteria. What does archaeal look like? And what does eukaryotic look like? Well, let's take a quick dive. I mentioned they're different. So this is our gram-negative bacteria. This is what we've been talking about. Again, that conserve structure, moat B and Flig, right? That's in direct contrast to our archaea, which, as you can see, have a very, very different structure. Very, very, very different, right? And we'll talk about how archaea power them. But archaea rotate to power their cells forward, but they actually don't have any molecular similarity to bacterial flagella. So what this tells us is that the ancestor of bacteria and archaea likely did not have a flagella, and once these two groups split from each other in evolutionary time, they both independently evolved movement. So we'll talk about this in a moment, but the key here is that our bacteria are powered by proton motive force. So that's how they generate energy to power their flagella, whereas our archaea are powered by ATP. So two very, very different structures. And this is in contrast to our eukaryotic. So we have what are called cilium, and these are like little, they vary. So they can be like hairy coats on top of a microscopic eukaryote, or they can be a single cellular one. But as you can see, they're essentially like I don't know, like little pins that stick out from a eukaryotic cell, and they're comprised of these what they're called anaxinomes, and they look essentially like I don't know. I think they kind of look like a tomato, like a cross section of a tomato. Um, but the sort of specifics here aren't all that important. Just wanted to show you how the differences in between the three categories. But the big difference here that you want to take away is archaea is powered by ATP, and our bacteria are powered by proton motive force. And so when we're thinking of how this movement actually occurs, so our bacteria and archaea operate via rotation, spinning. Now, eukaryotics typically waving back and forth, like wagging like a tail. Now, this, I will make mention of one thing. So when we think of movement in eukaryotic cells, it can either be like a microscopic eukaryote moving, or we can think of it like a gamete, like a sperm movement. And so historically speaking, we actually thought that sperm and many other eukaryotes wagged back and forth. Think of like a dog's tail, right? Or think about like, like a fin in water. Actually, some brand new research that just came out actually shows that flagella in sperm actually operate via rotation. So they do spin like a corkscrew, very much like bacteria and archaea. So just as a note, but if you have the vast majority of eukaryotes actually do this wagging back and forth. But again, sperm, human sperm, any animal sperm is actually physical rotation like a bacterial and archaea flagella. As I mentioned, they're powered differently. So proton motive force for bacteria, archaea is powered by ATP. And eukaryotic pili and flagella operate the same way with similar structures, but the difference is length. So flagella are very, very long, whereas cilia are very short. And bacteria and archaea can have flagella at various different types, lengths, and orientations along their cellular um, bodies. So you could be what's called atrichous, which is the possession of no flagella. You could be monotrichous, which is one flagella at one end. You could be amphitrichous, which is one flagella at each end. Lophotrichous, having a bunch of flagella at one end. Or my favorite, because it looks like, looks like a hairy bacteria, it's periodotrichous, which is flagella all over. But at the end of the day, they all work the exact same way. They spin clockwise or counterclockwise to direct the movement. Before we go on and talk a little bit more about motility, there is other types of mobility that we do observe. And the one that's actually kind of the coolest is from a group of bacteria called the spirochetes. And you might have never have heard of spirochetes before, but one spirochete you definitely have heard of, and you didn't know it was a spirochete, is actually the bacteria that causes syphilis, so Treponema pallidum, it is a spirochete, and a spirochete is essentially like a corkscrew-shaped bacteria. Now, the way they physically swim is actually kind of interesting. They are powered by flagella, but the weird thing is their flagella are located in their membranes. So what does this look like? So we have this, what's called a protoplasmic cylinder. That's their inner cell. We have the periplasm, that outer membrane, and you can see the flagella just is essentially corkscrewed around. And what ends up happening is the flagella spin, causing the bacteria to spin, which causes essentially like a corkscrew-like movement through the liquid. It's actually kind of cool. Weird, but pretty cool. Um, in addition, we have another type of mobility where we have bacteria will use what is called a pili or a pilus. It's very distinct from flagella. It's just, oops, sorry about that. It's just a protein-like structure that sticks out. We're not going to get into the structural specifics of it, just to let you know this type of mobility exists. They have this pilus, what they essentially extend, attach to a surface, and essentially drag themselves along. And this type of movement, you see this actually in Staphylococcus aureus. It's called gliding. Other bacteria, such as flavobacteriums, which are actually really common industrially used bacteria, they use it to twitch. So they essentially extend the pills to help pull themselves, and like they do it really fast, so it looks like they're just kind of twitching around when you look under the microscope. But that's essentially what's going on here. So that's the other types of mobility. You can spin like a spirochete, or you can twitch or glide like with a pilus. So what are your advantages to motility? Why do you want to be motile? As you can imagine, investing in the energy to make a flagella is a lot, right? To make that protein structure, to make all those proteins, it's a lot of protein. Now, with that said, what's the benefits of it, right? If you're investing all this energy, what's the benefits of being motile? Well, the first and the obvious one is you can escape predation, right? If something's trying to eat you and you can swim away, well, that allows you to escape predation, right? If you think about the example I gave earlier in class of the tiger, right? If the tiger's chasing like an antelope or something, if that antelope is faster than the tiger, well, it could potentially not be eaten by the tiger. So it's a pretty big advantage. Or the converse is you could be like a cheetah, right? Being very motile like a cheetah allows you to hunt down and chase prey. So to put this in perspective, there's a bacteria out there called Delovibrio bacteriovorus. It's a bacterial parasite. And what it does is it consumes other bacteria. And Delovibrio, it swims at about 160 microns per second. And what it does is it likes to hunt other bacteria. In particular, it likes to hunt gut bacteria like E. coli. And most gut bacteria can only swim between 25 and 35 microns per second. But we have our Delovibrio that can swim 160 microns per second. So it can easily hunt down and kill E. coli, which is pretty cool, right? Its motility gives it its capacity to eat other bacteria. So pretty fun. In addition, it allows you to move to a better environment. So if things are too hot, if they're too acidic, if there's not enough food, being motile allows you to move. But what are the disadvantages of motility? Well, it's costly. It's both requires resources to build the flagella, and it is very energy expensive to do, right? Think about the cheetah, right? A cheetah can run 60 miles an hour, but it has to have all that muscle to do so. So it requires energy to build the muscle, but to operate that muscle requires an immense amount of energy. So it is costly. It also means you typically live a solitary life, meaning you're all by yourself. You're not living in cohorts with other bacteria. And as we'll discuss later in the semester, bacteria living together is kind of a big deal for them. It provides a pretty large evolutionary as well as competitive advantage when they can live and interact with one another. But just to show you what Delovibrio looks like, it has a host. It swims really, really fast, stabs the host, gets inside the host, reprograms the host to make more Delovibrio, killing and eating the host in the process, then making more Delovibrio. So it's a pretty cool bacteria. If anyone wanted to do their microbe wiki on this guy, I would be totally okay with it because it is a fascinating bacteria to read about personally. But so as I mentioned, one of the benefits of movement is to move to a better environment. And one of the main reasons bacteria are going to move to another environment is in response to a chemical. And this chemical can be a signal, it could be a toxin, it could be an antibiotic, or it could be food. And so what this does is it leads us to this topic of chemotaxis, which is directed movement in response to attractants and repellents. And attractants being something sweet or, think about like, if you have my wife is downstairs cooking a pie, well, that's an attractant to me. I'm going to go smell that pie and go towards that pie. Now, if my wife is cooking with vinegar, I hate the smell of vinegar. That's a repellent to me. So I'm going to get as far away from that vinegar smell as I can. The same thing for bacteria. If they sense something in the environment, whether it's good or bad, they're going to move away or towards it. And it just doesn't have to be chemicals. It can be other things such as light, oxygen, and temperature. And it is ultimately sensed by chemoreceptors, and these are special proteins that span the cellular membrane that allow a bacteria to sense the environment around them. As we mentioned, bacteria have to be able to respond rapidly to their environment. They're single cellular. If they stay in an environment too long, they risk dying out. So they have to respond fast. And one of the ways they respond fast is these chemoreceptors. So let's talk about this idea of chemotaxis. So we have what's called the random walk process. Now, I mentioned bacteria have directed movement, but that's only partially true. And I say it's only partially true because of the stupid way they've evolved to, to move towards an attractant. So the way this works is we have a hypothetical bacteria here on the upper left-hand corner, number A, or letter A, I should say, and it has chemoreceptors, and it's designed to sense an attractant. Now, pretend it has an attractant over here, and it senses that attractant. Well, it senses the attractant with these chemoreceptors, and it's like, oh, I want to go towards that. So we could pretend this is food. Let's pretend this is a big pile of sugar, and the bacteria is like, ooh, I'm going to go eat that stuff because that smells delicious. This is the pie for the bacteria in the case of meat. Now, what it's going to do to move itself forward, it's going to spin its flagella counterclockwise. Remember, counterclockwise motion propels you forward. And that's in contrast to clockwise movement, causes you to either tumble or stop forward motion. So what ends up happening? The bacteria senses things. It swims towards the attractant, spinning counterclockwise, stops by spinning its flagella clockwise, which causes it to tumble, so it reorients itself, and then it moves forward again, so it runs, it stops, tumbles again, runs, stops, tumbles again, runs, stops, tumbles again, and it repeats this process over and over again until it makes it to its attractant. But it's just the way the bacteria do things. They can't just stop their movement, right? They don't have brakes, right? They don't have, they don't use friction because they're in water, right? They don't use friction to stop like say a car brakes would, right? They have to stop somehow. And the only way they can stop is by spinning their flagella clockwise. But spinning them clockwise not only stops them, it causes them to tumble. And so what they have to do is stop, cause them to tumble. It'll potentially reorient their receptors in a different way. So they're going to run again so that they can tumble to try to face back the other way. So it's random walking. It's directed, but not quite the way as like my wife is cooking a pie downstairs. I'm going to go down there and eat that pie, right? Because I can walk directly to the pie. Bacteria don't quite do that. As we mentioned, attractants are what bacteria are trying to go to, but they can also be drawn away from repellents. And so what ends up happening is that an attractant actually causes a bacteria to sense it and tells the bacteria to spin the flagella counterclockwise. And so if we have a bacteria that has multiple flagella, it actually causes the flagella to bunch together and spin counterclockwise, pushing the bacteria cell forward. This is the run. This is the straight line movement. Repellents do the exact opposite. They cause the flagella to fly apart, cause the flagella to spin clockwise, and causing the bacteria to tumble or change directions. But chemotaxis, again, as I mentioned, is a process of run and tumbling, causing that random walk. But what attractants do differently from than repellents is the receptors actually that detect attractants, they cause the increase of the frequency and the time period of a run. So when a bacteria is sensing an attractant, it runs longer. It runs in a much more directed process, just as a note. That's in direct contrast to repellent. So if you have a repellent present, it increases the frequency of tumbles. So increases the frequency of reorienting the bacteria away from the repellent. So, but there is a sort of overall mechanism here to how this works at the molecular level. And so we have our receptor protein on the outer membrane, and the way, the inner membrane, and we have our flagella, right? So outside of the cell, inside of the cell, well, periplasm, so in between the membranes. And then we have this receptor protein. And now the way this ultimately works is if there's no attractant, we're causing tumbles. So what ends up happening, we have this whole protein complex. And when the attractant is present, what ends up happening is CHEY or CHEY is phosphorylated. And when CHE is phosphorylated, it then, once it's phosphorylated by this protein complex, it causes a conformational change in the flagella protein to cause it to essentially spin clockwise. That's in direct opposite to an attractant present. So the attractant is bound to this protein complex, and it causes CHEY to be dephosphorylated, which then causes which causes a conformational change in this protein complex here, which causes the flagella to spin counterclockwise, causing a run. And you're gonna have to give me one moment. My dog is barking again. I really apologize. Just give me one. Okay, so sorry about that again. I gotta do these recordings when my wife is home. All right, so back to where we left off. So presence and absence of attractants causes changes in conformational proteins in these receptors to cause different changes in the spinning of our flagella. Now, we can actually look at our chemotaxis proteins, so our CHEY and CHEY phosphorylated. So remember, CHEY is involved in the receptors, involved in the response to an attractant, and we have blue, which are flagellar motor proteins, and we have CHEY unphosphorylated in black. And you can see these are located all over the cell. So the cell isn't just, I think it might have been a little bit too much simplification here. They're not just located at one area. Receptors for chemical attractants are located all over the cell. So bacteria has a lot of receptors because remember, you have to be able to sense your environment. And that's not just true for bacteria, that's true for you as well. You don't think about it, but you have the capacity to sense your environment all over your body. So with that over, let's just summarize what we talked about this first part that was only interrupted twice by my dog. I do apologize. He is a puppy, so he's kind of a pain in the butt, but he's really cute, as I showed you in that I think in that very first lecture slide. So the cell wall is structurally strong but porous. The cell morphology is important and depends on the presence or absence of key enzymes. Flagella are the main method of microbial motility, but their structure and power source varies among microbes. Remember, bacteria, it's about proton motive force. Archaea, it's about ATP. And eukaryotes, it's one or the other. Direct movement towards or away from a stimulus involves a random walk process that results from directing the flagella to rotate either clockwise or counterclockwise. Remember, clockwise causes a tumble or a stopping of forward movement, and counterclockwise causes a run or moving forward in a straight line. Um, not the not the smartest way to move, but that's just the way bacteria do things. They don't have what we have, right? And they're always in a liquid environment. Bacteria don't exist in, non-liquid environments in terms of movement. They can't just swim on a solid surface. They have to be in liquid to do this. So, all right. So next up in our, this is sort of just a two-part class today. It's going to be talking about microbial growth. So the questions we're going to try to answer are, how do microbes grow, divide, and reproduce? And what are the phases of growth? But let's talk a little bit about nutrition. Because growth is directly tied to nutrition, right? And as we mentioned in the very first class, bacteria are metabolically diverse, meaning they can use lots of different things for energy sources. And again, that is in direct contrast to eukaryotes, which are metabolically not diverse, meaning they can only use a very limited number of food sources and electron acceptors. So we can classify a bacteria based upon its metabolism. And this classification is based upon three characteristics. The first is the energy source. So how are they generating their ATP? Second, what is their carbon source? What does the organism use for carbon? So what food are they eating? And third is, what is their electron source? What supplies the electrons to power its electron transport system? Because when we think about metabolism, it's all about electron transport systems. Well, everything but fermentation is based upon electron transfer systems. But we'll talk about this metabolism in a couple classes. Again, the class you're going to hate, but we're going to talk about it anyways because it's really important. So what do I mean by energy sources? Well, how do you generate ATP? you should remember back from intro bio that ATP, or adenosine triphosphate, is the energy source for all life on this planet. Right? What powers your cells is not food, it's the food being converted into ATP, which is the chemical energy of cells. And so there's two different ways you can get ATP. The first is either as a phototroph, so you're performing photosynthesis, or you're as a chemotroph or a heterotroph, meaning you're taking external molecules, so sugars or fats or proteins, to make your ATP, whereas a phototroph is again taking light to make their ATP. Second is your carbon source. Where do you get it from? So if you're an autotroph, you get it from CO2, right? You fix carbon from the atmosphere to make sugars. Or you're a heterotroph. You take organic matter, sugars, proteins, fats, and you take them into your cell and you break them down. That's the difference in carbon sources. So autotroph or heterotroph. And then finally, you have your electron sources. These are different trophic groups. What do I mean by that? Well, you could be a lithotroph. You get them from inorganic molecules, meaning like nitrogen, metals, things like that. Or an organotroph, meaning you get it from organic molecules. And anything that's organic, it just is a carbon-based molecule. That's all. And we're going to talk about this much more in depth later in the semester, but just a sort of a foreshadow here. What do I mean by electron transport systems? The idea being is you have some sort of electron donor that gets oxidized. And there's a series of reactions along proteins in the membrane that essentially push protons across the membrane to create a proton gradient. So this is very similar to that proton motive force that I mentioned for how you power a flagella. As you get to the very end of this electron transport chain, it receives the final electron, and what it does is it causes the proton gradient to essentially um, uh, go through a protein called ATP synthase, which is basically like a rotor. Think of it like a hydroelectric dam, when water throwing fluid spinning a turbine. That's all ATP synthase is. And what this does is it converts ADP to ATP. So ATP being that chemical molecule. Now, the electron donor so the very starting process here, the electron donor that initially starts this chain to, again, generate all this energy. If it's organic or carbon-based, it's organo. If its electron is inorganic, like nitrogen or a metal, it's litho. And so the major trophic groups we could talk about is energy source. So again, photo versus chemo. Phototrophs are light absorption, whereas chemotrophs are chemical absorption of energy. Electron source, we have litho, so it's inorganic molecules, or organo, which is organic molecules. And then where do you get your carbon source? Auto, if you're using CO2 from the atmosphere, or hetero, if you're using preformed organic molecules. And microbes typically fall into 5 categories. So you could be a photolithotroph, which means you have CO2 as your carbon source, light as your energy source, and you're getting your electrons from an inorganic source. And again, inorganic sources are things that are not carbon-based. We have photoorganoheterotrophs, where they get their carbon for organic carbon, so food from the environment. They get their energy from light, and they get their electron source from organic electron donor. We have chemolithoautotrophs, where they get their carbon from CO2. They get their energy source from inorganic chemicals, such as sulfur, and they get their electrons from inorganic things. We have chemolithoheterotrophs, which, and I'll just talk about these next. So chemolithoheterotrophs and chemoorganoheterotrophs, they get organic carbon, and they get their energy either from inorganic chemicals or organic chemicals, and then they get their energy electrons from inorganic versus organic electron donors. And so what we're seeing here is metabolism of microbes is very diverse. So these are the 5 major categories. All these categories only exist in microbes. They don't exist in eukaryotes at all. They just exist in microbes. So, and there are exceptions here. So there are some microbes that are what we call mixotrophic or mixotrophs, the microbes that have the capacity to use several different carbon, electron, and energy sources, and they're not limited to one of these 5 categories. And what they do is, depending on the environment, they will change their trophic designation depending on what's available. So this is something that's widespread. You've seen this in some eukaryotes, but it's very, very common in soil, and it's very, very common in the ocean. And one of the ways that we know this as best is actually through study of the ocean. So we know this from looking at plankton in the ocean in Antarctica. So it's a pretty well-known thing. but it's actually kind of a cool thing. So I actually want to just briefly discuss this because I think it's a really fun paper. So mixotrophy, a widespread and important ecological strategy for planktonic and sea ice nanoflagellates in the Ross Sea. Nanoflagellates are microscopic eukaryotic organisms. And so these are phagotrophic, very much like your white blood cells. They will engulf and eat bacteria. And it's kind of cool. So they will eat bacteria, consuming them, but they are also chloroplastitic protists, meaning that they're algae, meaning that they have the capacity to photosynthesize. And it's kind of cool. So when the bacteria are present, they'll eat them. When there's not enough bacteria present for them to sort of hunt in a meaningful way, they'll switch to photosynthesis. But the cool thing is they combine the nutrients they get from the bacteria with their photosynthetic apparatus to combine them together into one metabolism. And it's actually pretty cool. So they're a significant source as primary producers and consumers. So in this area of the Ross Sea, where this paper is from, they actually contribute up to 50% of the total photosynthesis and up to 80% of the bacterovory or eating of bacteria in this area. So this is a eukaryote doing it, but just to know that there are bacteria that do the same as well. In addition, there's actually another cool type of bacteria called Rhodopseudomonas, pretty cool bacteria. And it changes depending on whether it's in the presence of oxygen or in the absence of oxygen. So it's a really cool bacteria. So let's talk about this bacteria just really quickly, and then we'll move on to talking about growth. But this guy's cool. I like marine microbiology. This guy's a marine microbe. Now, when it's in aerobic condition, it's chemoheterotrophic. What does that mean? Well, it's using chemicals. In particular, it's using sulfur. In this case, it's thiosulfate in hydrogen to break down organic molecules to generate its energy. So chemo, heterotrophic, it's eating stuff and it's using chemicals to get its energy and its electrons from. That's when oxygen is present. When oxygen is absent, it switches to photoheterotrophy. And what does that mean? Well, it uses the sunlight to generate electrons for itself, but it's still consuming organic matter. So it's using the electrons generated from solar energy to help it break down organic matter. So pretty cool guy. It's what we call a purple non-sulfur bacteria. Pretty cool. If you wanted to do your microbe wiki on this guy, I'm all for that as well. I think it's a fascinating bacteria. But again, I'm kind of biased because I like marine microbiology. So let's move on. Let's talk about growth. So microbial growth is based upon this idea of how microbes are growing from single cell to many, 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 many cells. Remember, prokaryotes are always single cellular, but there can be a lot of them in a given area. So as I mentioned, you have 38 trillion bacteria that live on and inside you. But all those bacteria had to start somewhere. And they had to start from a single bacteria that grew into a bigger population. So when we're in the lab, if we were in the lab together, we would grow bacteria on petri dishes. So all the petri dishes is just a plastic dish, and what it's filled with is, you can see this like jelly-like substance, and it's actually kind of a little bit harder than Jello. And this is agar, and this agar contains two things. It's a physical surface for the bacteria to attach to, and it has lots of nutrients for the bacteria to eat. So it's And the idea being is anytime you see a single colony of bacteria, single bacteria landed there and reproduced exponentially to produce this colony. So all a colony is just a bunch of bacteria living on top of each other, living beside one another. And again, it's all based upon the idea that it's clonal. One bacteria landed there, reproduced millions of times to create this genetically identical colony of one bacteria. Now, this plate has well, you could argue three, but definitely two different bacteria on it. So you have white guy here and yellow guy here. But the idea being each one of these dots, each one of these colonies, one bacteria landed there and grew millions so you physically can see it. So they are clonal, they are genetically identical. That genetically identical part, as I mentioned here, is ideal for molecular biology. So if you're trying to study bacteria, having a large clonal population is important for understanding its genetics. And the question you should be asking yourself is, why are they clonally the same? Well, it's because of the way they reproduce. So let's talk about how bacteria reproduce. So we have a bacteria. It's at an early phase of its cellular cycle. So it's a single cell bacteria. So it has its cell wall, its cell membrane. It has a chromosome here in red, and it has its ribosomes, right? Very, very simple bacterial structure. Um, then as the bacteria sort of senses things are right in the environment, it starts to prepare for cellular division. So it gets longer. So FTSZ is involved here. It starts to reproduce its genome. Then what it does is it begins to essentially get longer and longer. It starts becoming two distinct cells. Now, um, our friend, I'm sorry, FTSZ would be starting to do this. MREB is the one that's elongating. I apologize for getting those confused. But FTSZ is starting to get this budding habit, and eventually it pinches off and causing two daughter cells. So this should look very familiar to you. This is what mitosis does, right, in eukaryotic cells, right? One cell replicates itself, its genome divides, becomes two cells. It's the same thing. It's just much, much simpler in bacteria. This is what we call binary fission, one cell becoming two. And again, it's very, very simple. It's involved in both MREB for elongation of the cell and FTSZ for essentially budding of the cell as well. And if you haven't sort of seen what this looks like under a microscope, I feel free to take a peek at this YouTube video. Watching YouTube videos over recording lectures is kind of weird, so I'll let you watch that on your own. It's a pretty cool YouTube video. It shows you what replication and binary fission looks like in a microscopic organism. So the idea being is once the cells are starting to get ready to grow and divide, so once they double their contents, you have to essentially elongate yourself. And so this happens slightly differently in in cocci versus rods. So we have our cocci here on the left. The septum itself has to elongate, but it has to elongate essentially kind of looks like a peach, I guess you could say. You could say a butt, whatever you want to say it is. It has to slightly elongate, but it does have to pinch, but it doesn't do much elongation. That's in direct contrast to our bacilli, which do a lot of elongation. Again, and you have that splitting. But again, the septum itself has to be on the center. It has to contract to get that separation to do daughter cells. And as I mentioned, FTSZ is involved in the cellular division process. So as we mentioned, FTSZ is present in bacteria, archaea. It's also present in mitochondria and chloroplasts. So the way mitochondria and chloroplasts actually divide in the cellular cycle for eukaryotes is exactly the way we're going to talk about here. Just as a note, if your memory is a little fuzzy from intro bio about cellular division in eukaryotes, just know that chloroplasts and mitochondria do replicate independently of the cellular cycle, just as a note. So, but FTSZ, as we mentioned, is distantly related to tubulin and does function as a contractive ring that drives the cellular separation. So the idea being, when you have FTSZ, the protein filaments form what we call a Z ring at the site of cellular division. So in A and B here, it's the pink ring, and in black, it's in the figure down here, it's black. And it is something that is localized to the cytoplasmic membrane as a whole. Now, just as a note, the idea being here, what it's doing is it's essentially trying to push these 2 points together. That's what's really happening. It's like essentially, you can think of it like you have a knife on both sides kind of compressing in. That's what FTSZ is trying to do. That's what's going on here. But let's talk about the cell cycle in E. coli. It takes about 40 minutes for a typical E. coli to completely replicate its chromosome and about 20 minutes to prepare for another round of replication. But the kind of cool thing is it enjoys a 20-minute generation time at 37 degrees in the lab. So how is this possible? Well, let's talk about that. So it takes about, again, 30 minutes for it to completely replicate its chromosome and completely divide. But after 60 minutes, it's on 2 replication cycles. And then every 20 minutes, another one. Well, how is this possible? Well, after the DNA itself is replicated, about halfway around the chromosome, a second round of replication begins. So what it does is it actually overlaps DNA replication, thus halving the dividing time. What does that mean? Well, as it's starting to, as you see, it's got one cell, it does its chromosome. At about halfway through the division process, it starts to replicate its genome again. So after that initial division, all subsequent divisions are essentially two at the exact same time. So it's kind of fun, right? It can do something really, really in a short period of time by maximizing its efficiency. So if you like to work, if you've ever worked in a factory or anything like that, maximizing efficiency is what it's all about. So, and generation time is pretty variable. And so when we sort of have this table here, these are pretty common microorganisms. Um, and these are mostly, here we have mostly uh, pathogenic organisms. These are some pathogenic protists, and these are some uh, non-pathogenic fungi. Now, what you can see for these pathogenic organisms, they have temperatures that are about the same as the human body. They're about the same as the human body. And what you'll notice is that the generation time is very, very fast. So this is about a half hour, 45 minutes, almost a full hour, right? Pretty short replication times, right? And there seems to be an overall relationship with how fast you can replicate and how good of a pathogen you are. So bacteria like Staph aureus and Pseudomonas are really good pathogens, and they replicate really, really fast. Something like Treponema pallidum, which I mentioned when we talked about spirochete movement, is syphilis. Now, Treponema pallidum has a very, very long generation time. And so for those of you that know anything about syphilis, those that get syphilis, it takes a really long time for syphilis to sort of build up in your body, to see symptoms of a syphilis infection. So there does seem to be a relationship between your pathogenesis as well as how fast you can divide. Now, what you'll notice here is that we move from generation time in our bacteria down to our protists. Well, you see that generation time dramatically increases. So you're talking about a few hours at the bare minimum, upwards of 3 days at the maximum. To put this in perspective, the average human cell can divide once every 24 hours. So just as a note. And then finally, you'll see our yeasts, Saccharomyces cerevisiae. It's a budding yeast. This one makes bread, beer, and wine. Has about a 2-hour replication time. Monolina fruticola is just a non-yeast fungi. Has about a 30-hour generation time. So bacteria clearly are dividing much faster than the eukaryotes, but again, there are some eukaryotes that can replicate themselves really, really fast overall. And those are some pictures of what we're looking at here. So how do we model growth? Well, growth is not linear, and there are a limited number of generations a bacteria can have before its nutrients are consumed. No matter what environment an organism in, there is always a limited amount of nutrients present. So there's only so much of a population a given environment can expose. And so what we end up getting for bacteria is what we call the bacterial growth curve. And this is a pretty standard curve. Pretty much all microorganisms follow this curve. So we're going to go through this. We're going to talk about what's happening at each phase, but let's just give a brief overview of what's going on. So we have time on the x-axis here and log of the number of viable cells. So this is a log scale. And when I say viable cells, I mean cells that can be grown again on nutrient agar. So what you see here, we have 4 distinct phases. We have lag phase, we have the exponential phase, we have stationary phase, and then we have death phase. And it follows this predictable pattern. Now, the time frame here, this x-axis, varies depending on what organism you're looking at. So something that has a very, very long generation time, this curve is going to be pushed out much farther. But something that has a very, very short generation time, it's going to happen much quicker. So let's talk about the lag phase first. So in the lag phase, the cells are synthesizing new components, they're replenishing spent materials, they're adapting to their new medium. Now, this lag phase does vary in length. In some cases, it can essentially be instantaneous, or some cases it can be absent, or some cases it can be very, very long. It just depends. Becomes 4, 4 becomes 8, 8 becomes 16, and so on and so forth. That's exponential growth. And the interesting thing about exponential growth is that this rate of growth, so the amount of cells we're adding, is constant. So that's why it has this linear progression. In terms of the population as a whole, the population is the most uniform in terms of its chemical as well as physical properties. So the cells are all doing pretty much the same thing in this phase, and they're all pretty much comprised and have the same amount of nutrients inside them as well. Next up is the stationary phase, and this is where things plateau. So the total number of viable cells remains constant. And so this happens for a couple different reasons. Typically, it's because the cells have become metabolically inactive because they're starting to run out of nutrients. And so they're starting to run out of nutrients, or they're starting to sense they're running out of nutrients, and so they're going to stop reproducing. It can also happen because the reproductive rate is balanced by the death rate. And what does that mean? Well, it means that if you have 10 bacteria dying, well, that means 10 new bacteria are being born, which means you're not increasing the total number of cells here. So one of two things is happening there. And then finally, we have the death phase where the nutrients are completely exhausted and the cells are starting to die without replacement. And this is where we start to see the population decreasing over time. So we have these four distinct phases of our bacteria: lag phase, exponential phase, or log phase, stationary phase, and death phase. And this is consistent across all bacteria. This is consistent across archaea. This is consistent across yeasts. But this is, again, this is something we only see in microscopic organisms. This is not how you grow. This is not how fish grows. This is just pretty consistent to single cellular microscopic organisms, just as a note. And so what are some reproductive strategies that our microorganisms have? Well, for our bacteria and archaea, they're often haploid, meaning they only have half of the number of chromosomes that they typically need. They undergo asexual reproduction, again, no sex involved, and they're capable of what's called horizontal gene transfer. This is a process called conjugation. We'll talk about this process later in the semester. And the most common way they do this is by binary fission. This is in contrast to our eukaryotes. So both fungi and algae will reproduce by asexual haploid reproduction, but many eukaryotes have the capacity, such as yourself, for sexual reproduction. So these organisms will produce haploid gametes that fuse into a diploid gamete when the cell undergoes meiosis to regenerate haploid cells. Also as a note, protists such as ciliates here on the right reproduce sexually, but often form horizontal gene transfer to exchange genetic information. So they're doing basically what a bacteria do by having, it's not like traditional sex, but they're still exchanging genetic information. Remember, all sex is essentially changing genetic information. And just to sort of show you what this looks like, so we're going to look at Chlamydomonas. It's an algae. It reproduces asexually as haploid cells. Again, haploid meaning it has half the number of chromosome. So we're looking here, it has, so we have our mature cell here. And we see that what ends up happening is one of two things. It does haploid asexual reproduction, meaning it just reproduces more cells that have half the number of chromosomes, just like the way a bacteria would. One cell becomes multiple cells. So that's what it can do. Or alternatively, what it does is it generates gametes. So it generates these sex cells that are either plus or minus. And what they do is they fuse together. It can either be its own gametes or the gametes from another Chlamydomonas that fuse together to form a zygote that then immediately undergoes meiosis to produce haploid cells, which then turn into mature cells and either repeat this process over again, or they do the asexual reproduction. So just sort of an interesting life cycle. Remember, bacteria are always doing asexual. Eukaryotes have the capacity to do sexual or asexual reproduction. Just as a note, this is a really fun video. On YouTube, if you have a few moments to watch it. It's a little bit about reproduction in fungi. It's a pretty fun video with these crazy guys that are looking at measuring reproductive rates. And I mean measuring reproductive rates as in like how fast do these fungi shoot their spores, which is actually kind of a cool thing. So let's wrap this part up. So microbes vary in their sources of ATP, electrons, and carbon, and can be classified into trophic groups based upon these sources. Microbes use primarily binary fission, but also meiosis and mitosis to reproduce. Remember, our bacteria and archaea are always doing binary fission, but our eukaryotes have the capacity sometimes to switch between binary fission or sexual reproduction. Microbial growth does follow a predictable path through phases, and those phases are the lag phase, the exponential or log phase, stationary and death phase. And doubling times do vary considerably among microbes, and the length that these phases take does vary between microbes as well. And just as a note, those reproductive strategies do vary between microscopic organisms. So with that out of the way, that's going to be the end of class today. Please do let me know if you have any questions about anything, whether it's the syllabus, whether it's discussion, whether it's the material we just covered, whether it's anything going forward. Please do not hesitate to reach out to me. I am always here if you need to chat, whether it's by phone or whether it's by email or Zoom or anything like that. I hope you guys all have a great rest of your week and take care.